Hi, everyone. Okay, we're almost at 8 o'clock. I know we're eager to get started. Thank you so much for joining us tonight. And welcome to our newest webinar for OWNS. I am Dr. Neda Joya, president of OWNS, and we're excitedly kicking off September back to school with our latest and greatest Prevention is Better Than a Cure. It's a core message that resonates deeply within our mission. And at OWNS, we really believe that the key to optimal eye health lies in identifying potential complications upstream. So we really want to be proactive. And this aligns with the growing focus of prevention in healthcare. So tonight we have an informative webinar with Dr. Bruno called Mastering Functional Lab Testing for Optimal Eye Health, a deep dive into micronutrient and oxidative stress testing. Please use that Q&A button. We will be reviewing some questions at the very end. A reminder, this is a non-COPE webinar, and it will be recorded for free access. I know some of you have reached out. There are some competing things happening in about an hour. So even if you want to stick around for as much as you can circle back to us in our recording on our website. And usually we have that up about a week in time. Okay, so the Ocular Wellness and Nutrition Society is a registered 501c3. We're a nonprofit education organization. Our growth is really sustained through the contributions of our members, donors, industry supporters, sponsors, volunteers. By joining us as a member, you contribute to this momentum that drives our educational initiatives within the dynamic and evolving realm of nutrition and lifestyle medicine, especially in eye care. So if you don't know some of our newest features, we have Find a Practitioner. This is great to find colleagues. This has been already used by some patients out there looking for doctors who are really into nutrition or have some nutritional advanced degrees. We are continuing our amazing collaboration with the American Nutrition Association. We're one of the most exclusive organizations that offer you a 35% discount on the nutrition education. Please check this out. It is a very important part of educating ourselves to really get that foundation in nutrition. We are starting our own pilot, the ambassador program. I'm so excited about this. We're taking some applications currently. You'll find this on our website. This is open to volunteers, students, faculty, whomever wants to really engage into a more of a thought leadership role with OWNS. So please sign up. We will be having a private webinar for that soon. And webinars is what we do. We try to bring things that are a little bit different than what you get from classic COPEs and non-COPE webinars out there. So we're thrilled to bring two exciting new webinars coming at the end of September and in October. And the first is an in-depth session with Dr. Liz Lipsky. She's very well known, where we're going to explore various diets, introduce our brand new patient download tool. And we're also going to hear from Amy Smith and the ANA team about our collaborative education series, talking about that CPNP certification that I alluded to on the last slide. And to top it off, FullScripts will be offering some valuable insights onto how to grow your practice and boost revenue. Registration for this is completely free, and it can be found on our homepage, so please check it out. And then plus for our valued members, we have saved the date for our exclusive webinar with Dr. Jeffrey Anschel. He's our OWNS founder and former president, and he's going to share his insights about the complex condition of glaucoma and nutritional support. Remember, this is a members only webinar, so you have to log into your members benefit, and you will find that Zoom link there. So our initiatives don't stop there. We're continuing this Prevention is Better Than a Cure into our in-person meetup at Academy. We're thrilled to announce that we will have our awards reception at this year's Academy in Indiana RSVPs are now open and they will be on our website tomorrow, and you can sign up through our homepage or your membership benefits section. This year, we're celebrating an incredible milestone. We're welcoming 6 new fellows, which is our highest number to date in 1 year, and we'll also be awarding our $1,000 student poster prize and hosting a special student mixer, because that is our future, folks. The students have to learn about this to really change and help change healthcare in their futures. So save the date, and even if you can't attend the entire event, I still encourage you to join us when you can. The finalized agenda will be published next week, and we're looking forward to seeing as many of you as possible. I really personally believe that the power of in-person events, it's so essential to strengthen relationships and also to advance our mission. So I would love to meet each and every one of you individually. So I hope you can come to Indiana. So Vibrant Wellness, tonight's webinar, this is supported and sponsored by Vibrant Wellness. They're a leader in functional medicine testing, and their mission, our vision, is to understand the connectedness between your genetics, lifestyle, environment through testing, and provide personalized solutions for longevity. So it's a very unique lab testing company. I've used them personally, and I know we're going to have an awesome webinar about that tonight. And I would love to introduce next Dr. Kim Bruno. She received her doctorate in chiropractic medicine from the National University of Health Sciences in Chicago, and she successfully completed her CCN. She also continued her postgraduate education through Institute of Functional Medicine and Clinical Nutrition Certification Board A4M and ILADS. She's currently the Director of Clinical Outreach and Senior Medical Science Liaison for Vibrant Wellness Lab, and she's working to educate providers like yourselves on the use of functional medicine labs for treating complex patients. So without further ado, I am going to introduce the lovely Dr. Kim Bruno. I'm going to stop sharing and allow her to share her screen. So thank you for joining us, Dr. Bruno.
Thank you so much for having me. Thank you, Dr. Joya. Okay, I'm going to go ahead and share my screen as well. Thank you, everyone, for your attendance tonight. So today we're going to be talking about mastering functional lab testing, and specifically about oxidative stress and micronutrient status. We have a lot of tests at Vibrant, but those are the two that I think are going to be the most influential and nice additions into an ocular health space. And so we're going to go ahead and start with those. A lot of this we already touched on. Obviously, disclosure, I do work for Vibrant. Thank you so much for having me this evening. So who is Vibrant? We are a technology company at our core. We actually started as a technology microchip company, and then now are utilizing that type of technology to provide cutting edge, high quality technology with high amounts of accuracy, reproducibility, and comprehensive testing across the functional medicine space. We really are just fascinated with human health, all aspects of it, and how we can play a role to better give providers, and give patients the information to the upstream that Dr. Joya was talking about, right? It's one thing to pull people out of the river, but we really need to be asking why they're falling in. And that's really what we're going to be looking at as a lab. We are CLIA certification. We do have a CLIA certification and a CAP accreditation, which is going to be the highest level of certification and accreditations that you can get for a lab, so that you can really have a lot of confidence in the testing that you are going to be getting. Everything is very science and research driven. For first probably 5 years of our company, all of our resources went into research articles with scientists being able to be third-party published, more so than kind of flashy marketing. We really wanted to make sure that our technology was vetted and was published. And so we have research partners like Johns Hopkins, Mayo Clinic, Cleveland Clinic, to be able to demonstrate what our testing can bring to the market. So now, without further ado, let's dive into oxidative stress. So this presentation is really going to be kind of two separate sections, but you'll see how they will blend together, because when we talk about anything with functional medicine, at the root is going to be micronutrients, right? Because micronutrients are the information to get our biochemistry from point A to point B. So we're going to start with oxidative stress. We're going to go through some clinical associations, really how it applies, refresher on what is oxidative stress, how does that work within our bodies, within aging, within our eyes. I'm going to show you the test and all of the great features that that has, and then we're going to go through our micronutrient test as well. So I think it's really important when we start talking about a topic like oxidative stress is like, what's the root cause? What's the clinical conditions? What is the clinical associations here? Because we can talk about, kind of inflammation as this big thing or oxidative stress, but ultimately us as providers, we want to know, like, how does it apply to my patients? What is going to be walking into my office? So let's take a look at that. So it really is, it increases naturally with age. We'll go through that. Our bodies create a lot of oxidative stress. So therefore, many conditions that are associated with aging, whether it's systemic or whether it's in the eye, are going to have a direct relationship with oxidative stress and our body's ability to buffer or manage oxidative stress through the antioxidant defense system. So things like cardiovascular disease and diabetes, which also will have a big link into overall eye health. So those imbalances in the ROSs, or the oxidants, cause, too many oxidants, not enough of the antioxidants, are going to be causing that cellular damage. And that ultimately is going to increase mitochondrial damage, increase cell damage, which is going to increase tissue damage, which is going to increase overall organ damage and function. And so we need to talk about what's the damage that's happening, why is it happening, and then we also need to talk about that antioxidant defense system and the enzymes that are being used to maybe reduce some of those oxidants. And those two things together are going to really drive a lot of those chronic conditions. So as far as clinical utility in mechanisms of action, and I realize that these two are not directly eye health, but I do have some that are going to be directly eye health. But fundamentally, you are seeing patients who are coming in with cardiovascular conditions and autoimmune conditions. When they fill out their paperwork, they're checking the box, right? They have some sort of cardiovascular issue and or autoimmune condition. And so at the root, those really come down to this mechanism of oxidative stress. On cardiovascular cells, it's really going to be triggering this endothelial cell proliferation, which is going to be causing some vasoconstriction, endothelial dysfunction, which is going to progress ultimately to atherosclerosis. And on the autoimmune side, we're looking mostly at kind of protein oxidation and this post-translational modification of proteins, which ultimately is going to result in the formation of autoantibodies and the body being able to the immune system getting confused and going after self. So fundamentally, what's happening at the cellular level will drive these bigger disease states. So there's a great article that I found in preparation for this out of Redox Biology. This is the abstract here, really talking about the mechanisms of action of oxidative stress in ocular disease states. And a lot of the markers that they specifically talk about are actually the damage markers that we're going to be measuring. So wouldn't it be amazing if you could measure those markers, several months, several years ahead of the patient coming in and saying, that they were experiencing XYZ symptom. They were having dry eye. They were already progressed to diabetic retinopathy, something of that nature. So this is a great article. Definitely refer back to it. I pulled some of my favorite slides from it and just little snippets from the images. So this one specifically on diabetic retinopathy, looking at hyperglycemia, as it's going to really induce that mitochondrial function. And it produces more AGEs, those advanced glycation end products. It also causes lipid peroxidation. When you have these things, then that can ultimately end up in diabetic retinopathy. But if we can test ahead of time on a scale of, kind of normal, suboptimal, or sorry, sub-abnormal or abnormal of what's happening as far as their these end products that they're putting out, these AGEs or their markers of lipid peroxidation, we potentially could intervene sooner. Same with dry eye disease. So imbalance, our eyes are so inundated with environmental factors, air pollution, particulate matter, UV, all of those. So then they have this imbalance of the ROSs versus antioxidant defense system. Think about that as a teeter-totter, and we're going to go into depth on that here just in a little bit, but I wanted you to really see how it can relate back to kind of that function or that mechanism that's happening within the eye and can contribute to this very, very vicious cycle that patients can get on when they're developing this dry eye disease. Meibomian gland dysfunction. So some of the markers here, the 4-HNE and the 8-OHDG. So these are markers of, one is a marker of lipid peroxidation, the other one is a marker of DNA damage. And these are markers of oxidative stress that have shown to have an association here because the body is not able to work through this SOD enzyme, and we'll show that in the antioxidant defense system. But again, what if we could test those markers sooner and we could really start to intervene and quench that oxidative stress that our patient maybe would not be able, they wouldn't need to be pulled out of the river at the end because we would know that they would be falling in the river up at the top. So when you think about oxidative stress and inflammation, you want to think almost two sides of the coin, because for a long time in medicine, functional medicine in particular, right, we've been looking and paying attention so much to inflammation. Up until this test was launched about 6 months ago, there was really only one marker that we could really test for, one damage marker for oxidative stress. And for that reason, not a lot of people were looking at it. We kind of knew it was there. We learned about it in medical school. We knew that that could be a problem, but ultimately we were still always going after inflammation because we could measure it. We had a lot of inflammatory markers. So when you have a patient who presents kind of with an well-established inflammatory disorder or inflammatory signs and symptoms, but their inflammatory markers are within normal limits, or you work on inflammation with them and they don't respond to those expected treatments, I really want you to be thinking about oxidative stress and starting to go down the road of evaluating what their antioxidant defense system pathways are doing, as well as the damaged markers that are producing, in their system. It's very much a chicken or the egg, because inflammatory conditions can cause ROSs, ROSs can cause inflammation itself, and it kind of goes round and round. But if you're working in the inflammatory model and not addressing oxidative stress, you're really missing a piece for your patients. And this is a big piece, especially when we know that the eye has such high metabolic activity plus high amounts of direct insult from our environment, that oxidative stress is really probably causing majority of the issues, even more so the inflammation. Okay, so let's take a step back to medical school with me. I'm not going to go through all the nitty-gritty, but let's remember, what is oxidative stress, right? So reactive oxygen species are created, it attacks the cells. Ultimately, though, it really comes down to this teeter-totter effect that is going to have the impact of the genetics as well as the diet and the lifestyle, and lifestyle also meaning the environment. Because we've all seen patients who, are like, well, I well, they always comment, right, that they have this neighbor who's 100 years old who drinks and smokes and never wears sunscreen and is just healthy. And then we also have seen the patient who's like in their 20s and they look like they're rapidly aging and kind of falling apart at the seams. Some of that is going to come down to genetics, even independent of their lifestyle, and that has to do with that antioxidant defense system. And so when we are doing proper evaluation, we want to balance what's happening with their genetics and their antioxidants on one side and what's happening with their lifestyle on the other. And ultimately, are we balanced or do we have a lot of damaged products, a lot of extra ROSs that we need to look at? So there's two parts of the test that we're going to go through. You all remember free radicals. I always laugh at this slide because like chemistry was pretty much one of the classes that, got me to like almost quit going the medical route. But remember, we're missing a electron, which means that's why it's highly reactive. This is also why we don't measure free radicals directly. We've got to measure damaged products because they're so unstable that they're not things that we can measure in the blood. So how are those ROSs and RNSs formed? Well, the first two processes, normal cellular processes and inflammatory processes, these are within the body. And for the most part, everything is kept in balance, even if somebody has kind of faulty genetics for a certain amount of time. Our body knows that it's going to be creating those ROSs, and it has built-in protection to deal with it. Unless there's a situation where, like with an inflammatory process, you have somebody who has chronic Lyme, or you have some sort of situation where things are going unchecked. What really tips the scales is the oxidant exposure through diet, lifestyle, heavy metals, environmental toxins, UV, mycotoxins, all of those things. And that is going to be what's going to push it over the board. And if you have too much of that oxidant exposure, and then you also have, let's say, a chronic illness or a chronic pathogen that you're dealing with, now all of a sudden the normal cellular processes start to go a little haywire. So when we talk about normal cellular processes, when we're young, we have this like well-running car and it's got some exhaust, but not a big cloud of smoke coming out the end. But the more damage that those mitochondria become from the bucket being too full of the oxidant exposure, the more that your nice shiny car starts to turn into an old farm truck. And I couldn't find a farm truck picture. But just imagine, right? So now we have this older car that's kind of breaking down. We have a lot of exhaust coming out the back because those mitochondria are just not working efficiently. So this can increase as we age. It naturally does, and it's something that we have to account for. So 100,000 oxidative attacks on your mitochondrial DNA per day, and that's if your mitochondria are functioning properly. And how many patients do we see on a daily basis whose mitochondria are functioning properly? I know I don't see very many of them, right? Because we are ultimately seeing patients who have damaged their mitochondria for various different reasons. So therefore, the mitochondria, the free radical susceptibility to the eye, because it's so metabolically active, right? So it generates, the mitochondria is going to generate those ROSs. It's going to create the superoxide, which is going to create the oxidative damage, the mitochondrial DNA mutations. This creates the dysfunction. The dysfunction is the old farm truck now, but now as the exhaust comes out, we have more ROSs, and that impacts the eye. And now we're perpetuating all the way around. The same can be said for aging. The same can be said for other types of chronic conditions. But because of the number of mitochondria in the eye, we're going to see such a big effect with the oxidative damage or the reactive oxygen species that's happening. So that's your normal cellular process. So just through that, we're already putting ourselves at increased risk. Then we have inflammatory processes, right? So your innate immune system produces these ROSs to kill invading pathogens. But the example I gave before, what happens if the patient has chronic Lyme? What happens if the patient is living in a moldy house and they have mycotoxins? What happens if the patient continually is getting sick and their immune system is struggling and they're constantly creating these macrophages that are increasing more and more ROSs to try and kill pathogens? Remember this slide also, because when we talk about ages and we talk about those advanced glycation end products at the end, also remember that our macrophages sometimes are going after things that aren't necessarily a pathogen, but are foreign to the body, just like so the ages or those advanced glycation end products, those macrophages can go after and increase more of those ROSs as well. So we say here, we're at this point, we're like, so, okay, everyone gets an antioxidant. Well, your free radicals aren't always harmful, right? It's all about balance. We want some of our mitochondrial, some of our ROSs actually, help your body with antioxidant signaling and help to promote health. They help to like spur on the system, right? So that hormetic effect, like a little bit is great, but not so much. We don't want too much. So if you think about it in a kind of schematic here, you get this priming in this initial oxidative stress. So you don't want to completely shut off those first two processes. You want your macrophages to be able to respond. You want your mitochondria to be creating energy. So they're obviously going to be kicking out some ROSs. But what happens is we don't want it to spill over into inflammation. We don't want to kick off that NF-kappa B. And we also do not want it to fall over into apoptosis, where we're going to have massive amounts of oxidative stress, and that's going to be where we're going to get a lot of our cellular and tissue damage, because you're getting cell death and ultimately tissue damage and ultimately function damage. So we don't want to shut it off completely. And that's why it's important that we test, because we don't want to just like give everybody massive amounts of antioxidants. And I'm sure you guys have seen that too, is that if you give glutathione to the wrong person, right, they don't respond real well. So we want to be able to really customize this for them. So that all has to do with those kind of first two natural ways that we are creating oxidative stress. Now, this is your increased oxidants: too many calories, cigarette smoke, alcohol, UV radiation, environmental toxins. And all of our patients are exposed to majority of these things, right? They're all exposed to UV radiation, environmental toxins, heavy metals, mycotoxins, even if they don't want to be. I mean, we live on planet Earth. Maybe they can avoid cigarette smoking. Maybe they can avoid alcohol and eating too many calories. But majority of our patients here in the United States are going to be overfilling this bucket, even if they're trying not to. So we really are trying to figure out how we can get that down and how much damage is happening. How much capacity does their body have? So remember, when we talk about antioxidants, we're talking about things we can take, right? Things we can eat. So we can take vitamins, we can eat plant phytonutrients, but we also have endogenous antioxidants. And that's where I want to spend a lot of our time, because if we can dial in on our antioxidant defense system, the endogenous antioxidants that we can make and the pathways that they need to go down and also be recycled, then we have a big benefit to being able to help our patients. So here are your endogenous antioxidants. These are antioxidants that your body makes. They are going to either do one of two things: donate an electron, or they are going to help recycle another antioxidant so it can donate an electron. All right, so when we are looking at enzymatic, and that's going to be a lot in the pathway. And then the non-enzymatic are things that you can take some of these, and some of them are going to be things that our body is also going to be creating through the system. So this is a very simplified schematic of our antioxidant defense system, but this is our built-in protection, right? So we have your endogenous and exogenous triggers. which are going to be creating ultimately superoxide. And then we need to determine where it's going to go to clear out of the body or if it's going to get stuck. All right, so have that. There we go. So your superoxide can interact with polyunsaturated fatty acids. This happens a lot in the eye, right? There's a lot of fatty acids in the eye. If we don't have enough antioxidants, think about it as like those cookie sheets that like get that film on it, like because the oils all kind of crust onto the cookie sheet. So we need to be able to recycle, use our vitamin E's to be able to get this system back in order. It also could go down, your superoxide could go down the SOD pathways, 1, 2, or 3. This has to do, and this gets your antioxidants, or sorry, your superoxide to turn into hydrogen peroxide. Well, we don't want to stop there because hydrogen peroxide is also an oxidant, and it also can create oxidative stress in our system. So we still need to go down 2 more pathways to clear to water. So that's going to be 2 more enzymes. We have our glutathione and we have our catalase. Let's get down here. So we want to get to water, but every point along the way, there could be a genetic SNP, which is going to be a variation that could inhibit that enzymatic activity, or we don't have enough nutrients to be able to properly get us down these pathways. And then we get stuck and then we can't clear. So when we're looking at damaged products, yes, we want to look at what's happening in the environment in that bucket 3. We want to evaluate what types of infections are happening, their mitochondrial status, but we also want to look to say, how are they able to manage their antioxidant defense system so that we can get those endogenous and exogenous triggers all the way through superoxide, all the way down to water. All right, so let's look at this in action. So think about that teeter-totter that I talked about at the beginning. We are going to evaluate genetic predispositions. Are your antioxidants working? Meaning, can they donate an electron or can they help recycle another antioxidant who can donate an electron? A lot of that has to do with your genetic predispositions through your antioxidants defense system. On the other side, we're looking for the actual damaged markers of lipids, proteins, DNA, RNA. What else is happening? Do we already have a problem that we need to really intervene and figure out what's causing that damage? So the testing profile is going to be a saliva and a urine test. And this is great because patients can then do it at home. So it's an at-home collection. You don't have to have phlebotomy in your office or anything like that. If you really love technology, you can see we are using real-time PCR for our genetics, and we're using mass spec for our damaged markers. This is just a comprehensive list of our damaged markers, of our genetic SNPs, thank you. And we are gonna map this out for you, but I just want you to have a complete list, so in case you want a reference slide. And then this is a complete list of our damaged markers. Now, remember when we were talking about mechanism of action for the eye, we looked at things like the 4-HNE, we looked at the advanced glycation end products, these ages here. And we also have documentation for 8-OHDG. And we also know that a lot of protein oxidation can impact what's happening in the eye as well. So, genetics 101. This is going to be super speedy, but I'm going to give you kind of my, how do I explain genetics to my patients? Because we want to make sure that we are not inducing fear when we talk about genetics. It is just helping to give us some more information so that we can better manipulate the flow of their biochemistry. So when you see SNP classification. So a SNP is a single nucleotide polymorphism. It basically means that you are going to, you have a base pair substitution. And therefore, when that base pair substitution happens at a coding part of the DNA, it creates a different protein, potentially. So the wild type is the most common, or what is considered normal. So when you look at a result and you see wild, you see homozygous wild, that means that is the normal type or the most common. Usually does not come with a genetic risk. If you see a mutant or a variant, that usually means that it is happening less frequently, and it usually carries an elevated risk for the patient. So you've got your homozygous wild type. You've also got your and your homozygous mutant, which we just covered. And then you have your heterozygous. So heterozygous is going to mean that you have just one base pair substitution and you have a potentially partially elevated risk. It depends on the highway that you're on. And so the highway analogy is like this. Normal or your homozygous wild, all highways are open. So if you think of a biochemical pathway, all of them are open, they're flowing, your enzymes are working, you have the right nutrients to move your enzymes through, everything is working along the way, all the traffic is flowing. If you have a heterozygous, you have partially elevated, which means you have like one lane closed. Depending on how much traffic is on that road, this may or may not pose a risk of a traffic jam, right? If we don't have a lot going down this pathway, then our body might be able to manage it just fine. But the problem then lies that if we're feeding in too much. So let's think about this in the realm of oxidative stress. If we are bringing in too many toxins, too much alcohol, too many calories, and we have one lane closed, we may get a backup in the system. And then your elevated risk is going to be your homozygous mutant or variant, depending on how you report it out. And this usually means that almost all the lanes are closed. There's high amounts of traffic that need to go down this pathway just on normal physiological processes. And so therefore you get a backup in the system. And then you can have this kind of, huge amount of oxidative stress that is now just kind of continually breaking down those tissues within that system. So what you want to know is, can I create a genetic bypass? Okay, so I know what's happening with the highways, and this is how I tell my patients. You know what's happening with the highways, but who's used the Waze app, right? Or really any navigation app. And what do they tell you? They say, oh, this road is closed, better take this road over here. Is there an alternate highway? How much traffic is there? And could we go, if it's a highway analogy, could we go at a different time? Can we take some of that traffic away? Can we take out some of their toxins? Can we get them to eat less calories? How can we affect them? And then what are the enzyme cofactors, which are all micronutrients, which we're going to talk about at the end, that are going to move that traffic along, right? And do those enzymes have the micronutrients or the cofactors that they need to be able to move the traffic along? And sometimes, especially if you have those like heterozygous, where you have a possibly partially elevated risk, if we have good people, routing traffic and they have all of the nourishment they need, we can keep the highway flowing. So when you get the report, you're going to get all of these genes. Don't these look familiar? These are the genes that control the antioxidant defense system pathways. The SODs are the ones that got us from superoxide down to hydrogen peroxide. So here we have some of our SODs have some elevated risk, meaning there's some breakdown in the pathway to get down to hydrogen peroxide. So we're going to have a buildup of superoxide, especially if we have more endogenous and exogenous kind of triggers that are coming in up at the top, versus some of the other ones are normal. And then once we get down to hydrogen peroxide, we have to clear either by the GXP pathway or we had to clear down the CAT pathway. And in this particular patient, they have two SNPs that have to do with their cat genetics, right? And so we need to be able to kind of work around that so that they can, we either need to overly support their glutathione pathway over here, the GXP pathway, or we need to make sure that we're supporting in the particular enzymes to help them facilitate that cat pathway as well. Okay, so let's look at how this looks on a sample report. So there's various different sections on the sample report. You're going to get an actual build out of your antioxidant defense system. Then you're going to get your genetics, your oxidative damage markers, and then you're going to get a lot of additional information, which was super helpful for you and the patient. So when we are trying to get our free radicals to convert into more stable forms via this antioxidant pathways, we got to use these genetic pathways. So this is your Waze app, right? This is your navigation app. You're looking for, are there elevated SNPs that inhibits the proper flow and therefore are things that we need to intervene. So we can look at some basic nutrient support here for the GST enzyme if we need additional support here. And I'm going to reference it at the end, but we do have a very complete interpretive guide, which will give you a lot of these treatment action items of things that you can work on. So you can give them glutathione, but you can also give them other types of polyphenols that can also help to kind of move some of those free radicals down into more of those soluble extracellular toxins so they can be excreted through the system. If your superoxide goes down to your free radicals, this can enter this problem here. So we need to be able to have adequate amounts, again, of glutathione to help with donating our electrons to help to stabilize these free radicals. And we need to make sure that we are not, that we are protecting their lipids as much as possible. If we start to go down that kind of standard pathway, down the SOD pathway, trying to make hydrogen peroxide, we're going to have some nutritional support here for SOD2 and SOD3. We need things like alpha-lipoic acid, CoQ10, vitamin A, vitamin E. So as you can see, it's not all about glutathione. Glutathione's great. It's a master antioxidant. We love glutathione in the right patient, but this patient also needs some additional antioxidants to help things flow through their system. And then at the end, this poor patient had both the CAT enzyme and the GXP enzyme, both have genetic SNPs. So they need both selenium and glutathione. So this can help us really build a lot of the very specifics as far as the antioxidants to help them flow through their antioxidant defense system here. You will get a flow chart with some analyzing of the genes that will just tell you, exactly what it does in more of a verbal representation. If the chart itself is overwhelming for your patients, sometimes looking at biochemical pathways is just a little bit too much. And you're also going to get a list of all of the genes, what its risk association is, what its mutation is it an elevated risk, all in a summary page, plus you will get the interpretation. This is all on the report. So you do not need to be a geneticist or have an expertise in genetics when you can really apply it. And if you simplify it into kind of that navigation aspect with our patients and just help them kind of walk through, you can see that these are modifiable, right? We can't change the gene, but I can create a bypass, I can support the enzyme, I can choose to be able to take some of the traffic off of that pathway. And ultimately that gives the patient knowledge for a lifetime, but that gives them actionable items that they can do, and it's not a gloom and doom type situation. And that's really what we want in genetics. We're not giving kind of a traditional medical part of genetics where it is a disease state genetics, where we're letting somebody know about a certain disease condition that they have. We're just letting them know about some of the, susceptibility in their highways or their pathways in their system, and how we can help manipulate and change it for the better so that they have better outcomes in the long run. Okay, so when you look at the report, that's the genetic piece of it. That was the salivary piece. That's really has one side of the teeter-totter. But then you're also going to look at your damaged markers, and you're going to get them in each of those sections, the lipid peroxidation, the DNA damage, and so forth. So you can see here, you can see whether it's normal. You can also, if you're doing repeat testing, we've got previous result versus normal result or versus current result, which is helpful to be able to really monitor. You're going to look at those suboptimal levels. Let's see here. And that was your previous and current, which we mentioned before. Also note, these are all fairly new markers as far as within the world of functional medicine. 8-OHDG for a long time, you might have seen it on a urinary hormones test or an OAT test, an organic acid test. But this comprehensive amount of damaged markers is a new test to functional medicine. So we include also the interpretation right on the test. In addition, you can get the interpretation guide so that you can have that as a desk reference, but you're able to really see what is it impacting, what are some of the disease risks that are happening with that for that patient. You will get an oxidative damage score. So they take all 16 damaged markers and they're going to give you a damage score. This correlates with somebody's biological, their biologic age. So this helps patients to really look and say, oh, am I aging in a healthy way? Or am I aging in a more accelerated way, or a moderately accelerated or an accelerated aging? And I will tell you from a compliance perspective, if you have a patient whose given age is 55 and they have an oxidative stress profile of a 66-year-old, they are quite motivated to start to implement some of the dietary and lifestyle changes that you are looking at. And then you can track here and they can see, oh, okay, so I originally had a 79, I was at age 79 and now I'm at 62. So that is going to be somebody who's going to be making those improvements based on repeat testing. As far as the lab test, you also will get supplement recommendations, because you want to be able to give those targeted, not just glutathione for everyone. You want to make sure, okay, this patient needed glutathione, but they also needed selenium. They also needed vitamin C. They also needed vitamin E. And it's going to give those targeted information based on their damage markers and their genetic risks. So let's take a little bit more time to look at those damaged markers, just so you kind of conceptualize why we're looking at damaged markers. Remember, I said you can't measure actual free radicals because they are so unstable. So the 8-OHDG and some of the other markers of DNA damage, ultimately, in the guanine section, you have that superoxide, which is going to come in and it's going to impact that double bond at the guanine section. And it's going to create 8-OHDG, and your body, as it comes in to heal your DNA, it excretes the 8-OHDG as part of its repair process. So the more of that 8-OHDG that you have in your urine, your body is trying to actively repair its own DNA. And so that's why it is a damaged marker, or a DNA damaged marker. It has it's a biomarker of early disease detection. Now, this particular study has to do with cancer, atherosclerosis, and diabetes. The study that I gave you at the beginning also had to do with dry eye. And so you can see that there can be some there's going to be some connection over in your patients who systemically are maybe dealing with atherosclerosis or diabetes, but it's also affecting their eyes. As far as lipid peroxidation, the marker that we talked about before was the 4-HNE. But lots of lipid peroxidation happens in the eyes because of the high amount of fat content. And so we have your lipid peroxidation again. Think about that cookie sheet analogy that I gave you before. Your polyunsaturated fatty acids, arachidonic acid is very susceptible to free radical, superoxide free radical damage, and it creates this damaged product. And when that happens, then this can be the beginning mechanism of action for atherogenesis and also other types of atherosclerosis and problems in the cardiovascular system, which of course would then impact the eye. As far as our advanced glycation end products, this is the one that we talked about with diabetic retinopathy. So think about this analogy as far as you have glucose and it's overheating with oxidative stress, and it's creating this really kind of like caramelized sticky bun type situation. That is the advanced glycation end product. Well, what's the problem with an advanced glycation end product? Is that your macrophages look at it as a perceived threat, just the same way that it looks at it as a pathogen. And so therefore, it's going to increase the amount of ROSs. And so you get into this very vicious cycle that it's not just the sugar and the inflammation that's coming from the sugar, but it's that it's also creating and impacting to create these advanced glycation end products, which is then creating more ROSs, which is then creating more mitochondrial damage. So you can see how this can kind of just a feed forward mechanism for this patient. There's several of these markers that go together in what we call, marker clusters. So I just put in some studies there. I'm going to zip through these just because I want you to have the references. They're all referenced in the slides. You'll get copies of the slides, but patients who have hyperlipidemia, they are going to have more problems with, of course, some of the lipid peroxidation or the lipid oxidation. And this one is a protein oxidation marker. When we're talking about diabetes, we've got DNA damage, we've got advanced glycation end products. We also have other lipid oxidation that's happening. With Alzheimer's, we have a whole host of things, everything from protein oxidation, DNA damage, and ages. So we really need to understand that if our patients are coming in for an eye exam, but they also are having memory issues, getting some good testing for oxidative stress earlier can help us really understand the whole picture of what's going on for that patient. If you really want to take a deep dive into all of these different mechanisms, then its role in health as far as it's relating to aging, in the 2 years it took to develop this test, our scientists did a meta-analysis. It's actually out for peer review right now, but you can access the text through this QR code. And you can read up on it and nerd out over oxidative stress, because I know I did for a long time when this test was in research and development, so that we could really understand its role in functional medicine. So let's talk a little bit about some of the approaches for management, because remember, we want to give our patients antioxidants, but we don't want to give them just antioxidants. We want to figure out why they're falling in the river, not just pulling them out at the end. And we need to evaluate what's happening. So once you have an idea of how well their genetics are working in their antioxidant defense system and how many damaged products they have, it can help to guide you to decide if you need to dig deeper. Is this someone we just need to talk to about good lifestyle and maybe less alcohol or something of that nature? Or is this someone that we really need to dig a little bit deeper into something like chronic infections or their toxins, their heavy metals, environmental toxins, and things of that nature? So our patients are always on a continuum, right? Unfortunately, in the last 4 to 5 years, I see many more on the further side of illness than I ever did in my 17-year career up to this point. I just think that our patients are being inundated by so many things, whether it is our environment, our stress, our diets, everything else is, it's really just challenging for them to kind of stay above it. So understand that this can be a test for most of the patients who are walking in for you. You do want to really identify and treat the cause. And so that depends on how many damage markers do they have, which damage markers do they have, will lead us to say, again, do I just need to work with them, get a health coach to help them with lifestyle factors? Maybe we need to address metabolic conditions, or do I need to dig deeper into something else from like a toxic exposure that might be happening? Lots of great supplements for oxidative stress, but I will say this before we start talking about micronutrients is let's know what these, what the status of theirs are first. Let's know what their genetics are. Is there a backup in their pathway? Do they have enough of these nutrients to get them down those pathways, or do we need to be a little bit more strategic? Because we don't want to just get into kind of a polygreen pharmacy, right? Where we're just throwing a bunch of supplements at them the same way that, they might be getting a lot of prescriptions. We want to make sure that we're really creating this personalized approach of the types of supplements they're taking. And also remember that we can always upregulate their endogenous antioxidants response elements through utilizing a lot of herbs, spices, medicinal plants, and phytonutrients. So let food be thy medicine. Have our patients utilizing lots of fresh herbs and spices, having them, look at things like green tea and coffee phenols, and what patient doesn't, would love to drink some more coffee and have some more cocoa or chocolate. So utilize that as some of your tools as well. Modifiable risk or lifestyle factors, there's lots of them. So they can be from diet, exercise, sleep, stress, but they also need psychosocial support and relationships. This is something that you can build a program for. And I know Dr. Joya was talking about some programs that you guys have already going on, and this can be part of that for patients to be able to really support them either through a health coach or through somebody else in your office to help them really hone in on this. So obviously some of the dietary risks that we already talked about before, excess calories is a big one, but also any types of these ultra-processed foods, which we're hearing about everywhere, right? Ultra-processed foods are the cigarettes of, the 1980s. So we really need to look at these dietary risk factors as they pertain to our patients. And if we can just start with dietary, we can probably make a pretty big impact because they are eating all day long, right? So if we can implement some of these modifiable lifestyle factors, that can be really helpful. Here are some studies on different types of diet and their patterns with oxidative stress. Obviously, Western diets were ones associated with highest amounts of oxidative stress and inflammatory levels. Kind of a different plant-based diets, lower oxidative stress and inflammation, moderate Mediterranean diets, lower levels of lipid peroxidation and less oxidative DNA damage. And the DASH diet, interesting, lowered lipid peroxidation and helped to increase nitric oxide. I'm not saying one diet is perfect for another. I mean, at my root, I'm a nutritionist, but I also think that everybody is a biochemical individual and doesn't always fit into a box. But just kind of use these as a guideline that you can, help to manage oxidative stress through those dietary patterns. Exercise and movement. We all should be moving always, sedentary again is the new smoking. So make sure that your patients are moving, but do note that very vigorous intensity does generate some oxidative stress. So if you have a patient who is very, very active or is a weekend warrior or likes to do, 50-mile running races or something of that nature, and you want to do this test on them, I would have them kind of just do moderate exercise so that you can see like a baseline, unless you want to see how much damage is being produced by them doing those high intensity workouts. But understand that there can be some manipulation here because your body is going to create some of that as well. And if we have somebody who has high amounts of oxidative stress, maybe we need to tone back to just kind of movement, yoga, walking, instead of having them do those like HIIT workout types of activities. Sleep, sleep, sleep. If they're not sleeping, it's really hard to fix anything else. There's not enough supplements that we can take to really overcome inadequate amounts of sleep. So dig deep into your patient's sleep and any types of disturbances that might be causing for them. Deal with stress management. Again, they could have everything on point, and if they're super stressed out, we could be creating a lot of oxidative stress and just kind of spinning our wheels. So looking at breathing exercises, yoga, meditation, any type of behavioral therapy, evaluating what's going on in their life. Can they switch jobs? Can they, are there things that they can do to help to mitigate that overall oxidative stress load on their bodies? And then, of course, psychosocial support and relationships, which is going to be pretty closely related into the stress piece as well. But we saw a lot of this in the last, around COVID time, social isolation, loneliness, lack of social support, having very adverse effects on health. And consequently, it is quite damaging to your oxidative stress biomarkers. And so these are things that we need to be talking to our patients about. Okay, let's switch gears and talk about micronutrients here. Thank you so much for hanging in with me on this. This is a lot of information. And we, like I said, you'll get the slides and you can circle back to the recording. Okay, so why do micronutrients matter? Well, for all the reasons that we just talked about, because biochemistry matters, right? Because at its core, we can't move through any of these biochemical pathways, whether we're talking the antioxidant defense system or whether we're talking the Krebs cycle. We've got to have nutrients to move us through all of these pathways. It is the information that is telling our body and our pathways what to do. Right? So it is not just about energy, macronutrients, it is about micronutrients and the information that is provided to us through food and supplementation to tell our cells what to do. And unfortunately, our patients are not in a great situation for micronutrient status. So up to 92% of Americans have some sort of nutritional deficiency. We are in a situation in a society that is overfed and undernourished. They have a lot of the macronutrients, they have a lot of calories, but they don't have a lot of the micronutrients. And so therefore their cells are starving, right? They just don't have enough to get them through the pathway. So maybe they have fine looking genetics, but their nutrient status is not there to be able to push them through those pathways efficiently. And then you put onto that some extra cars on the highway, and now for sure we are not able to get our system in a fully functioning pathway. So just some stats there that you can refer back to or talk to your patients about. And nutrients, although we love nutrients, they do have a U-shaped curve, right? It's not all roses. There is a potential for toxicity for some nutrients. So it's best to test and not guess, and not just throw everyone on high levels of micronutrient or of a multivitamin. We want to be able to get them into this kind of upper end range, this optimal level, maybe that optimal daily intake versus the recommended daily allowance. But we need to be able to know how deficient they are so we can pull them up out of the trenches and then be able to maintain without going overboard, right? We do not want to cross over. There's definitely studies out there like too much vitamin D, right? We have people who are like, my vitamin D is whatever, 180. Like, okay, well, there's increased risk and potential toxicity, and we need to be able to really manage them in this U-shaped curve. So these are all the different nutrients that are going to be on the micronutrient test. And the big difference in what stands apart for Vibrant is that we are doing a direct measurement, not an indirect measurement like some of the other tests on the market, but this is a direct measurement of both intracellular and extracellular levels. And this is really important because extracellular nutrients, a basic, vitamin B12 or whatever that you're going to be getting at like LabCorp, these are reflective of a very short time frame because our nutrients, the biochemistry that I showed you before, it doesn't happen in the extracellular space. It happens intracellularly. So yes, we want to know how much they're bringing in through diet, through supplementation. That's your kind of extracellular, more transient, short time frame. But intracellularly, that is where they are transported either into the white blood cells, into the red blood cells. This is going to be really reflecting the nutritional status of the lifespan of a cell, which is where biochemistry happens. So it's helpful for us to be able to see kind of what is the cell function or the nutrients that are happening within the cell function if we're testing the intracellular levels. And we want a direct measurement of that, not an implied measurement. We also, because we are testing cellular levels intracellularly, we are going to report out your white blood cell levels because depending on what's happening with our patients, and sometimes our patients have low white blood cell counts or high white blood cell counts, it can create a situation where, like with low white blood cells, it can falsely kind of concentrate those analytes and give us some higher levels. Or with high white blood cells, it can dilute the analytes and give us some false lows. It does not invalidate the test, but you want the calibration, and any lab test that's looking at intracellular micronutrients should be reporting total cell counts to you. So you as the provider can have that level of calibration so that you can say, oh, okay, they had low amounts of white blood cells totally, and I'm seeing a bunch of things at the upper range. Those might be a little bit falsely high. They might be more mid-range. And you can just kind of clinically do that. Here's some possible causes of those abnormal white blood cells. Just for your reference, you can look back at them in case you have a patient whose white blood cells are off. Remember to look for patterns in your testing because depending on the types of patterns, the types of nutrients that are deficient, you may need to think about gut health. And because it's not so much that you are what you eat, you are what you can digest and absorb. So we can't really talk about nutrients unless we also remember that all of the digestion and absorption is happening in the GI system. And if you start to see certain patterns of nutrients being low, you might want to take a deeper dive and looking into the GI system and its function there. So when you look at the clinical interpretation, you want to make sure that you're asking your patients about things that happen in their lifestyle, diet, stress levels within kind of up from the last week, month, and 3 months. And that is because the serum, which is your extracellular value, is good for about hours, days, maybe up to a week, right? So if this person ate liver the night before they came in to get their blood drawn, they might have some nutrients that are higher than before in the serum. But in the white blood cells or in the red blood cells, you may see, like now you might see this discrepancy that they have really high serum levels, but kind of mid-range in their intracellular levels. So you want to make sure that you are knowing kind of like, did you have a typical diet? Did you have any stressful events? Did you run a marathon? What types of things are happening at those time frames so that can help you get some ideas around their utilization of nutrients. You're going to get these dial charts. And so these dial charts help you to see clusters of nutrients that pertain to different systems in the body, right? The most common, our patients understand that like vitamin C is good for the immune system, but they don't really understand that like vitamin D and iron are good for your bones and joints and muscle health, or maybe your vitamin E and your iron and your EPA are good for your cardiovascular health. So based on the research, the lab has clustered certain nutrients together, and if they're low, it shifts on the dial. This is really just helpful for patients. Patients want to see all green dials. They want to see that the nutrients that they have are supporting their cardiovascular health, are supporting their liver detoxification, are supporting their mitochondria. So this is not a disease causing situation, right? Just because you have a low value for mitochondrial function doesn't mean that you have mitochondrial damage. But what it's saying is you don't have all of the nutrients needed for proper mitochondrial function. So therefore, you're a higher susceptibility of aging. And so think about it in the realm of nutrients supporting systems. And then you can start to prioritize, do we need to do more testing? Do we need to do, a gut protocol? Do we need to really dig deeper into their cardiovascular system, or what might be happening there? So when you look at the results, now this is just a snapshot of the vitamins. You'll see that you have every vitamin listed, and then you have their serum result and your cellular result. So you have your extracellular and your intracellular levels. One thing you want to do at first glance is kind of see, is there a pattern? Well, the first thing I see with this patient is, yes, all these things are green, except for these two that are red, but most everything is green, but it's all like skating in that lower quadrant, right? Like they don't have any really, the only one that I would say they have kind of an optimal level of is their serum level of vitamin C. But their cellular level is still, they don't have enough reserves here. So these are things that we want to look at just across the board. This would clue me in to this patient is not digesting and absorbing the nutrients as effectively, right? They've burned through their nutrients for whatever reason. And now we're going to go backwards and we're going to talk to them. What happened in the last week? What happened in the last month? What happened in the last 3 months? What happened that you are not utilizing your nutrients? And they're going to tell us a story, and we're going to listen to them, and then we're going to start to build out a plan. You want to ask these 5 questions, right? What are the intakes of all their micronutrients from diet and supplements? What are they losing micronutrients, right? A lot from the GI system, but also detox pathways, urinary tract, skin. What else is happening? Do they have a skin condition that they are rapidly having skin turnover and they're maybe utilizing a lot of those nutrients? Is there a reason, again, for the utilization of micronutrients? Is there a cellular transport problem? This comes down to your omega-3 fatty acids, because if you have higher amounts of arachidonic acid and lower amounts of EPA, or your omega-3s, you have a stiffer cell membrane and there may be a transport problem. So you may have an okay level of serum, but not a very good level of intracellular. And then we always want to be thinking genetics as well. Is there potentially a genetic polymorphism? And that is called nutrigenetics, right? Where they are not able to convert, they're not able to process their nutrient as effectively. And maybe at a later date, we can talk about different tests that you can look at for nutrient genetics, which are separate than the ones that we talked about with the antioxidant defense system, but also really important to why and how we are utilizing our nutrients and transporting them and assimilating them in our systems. This is a little snapshot of your fatty acids. So like I said, like if this person has, arachidonic acid to EPA, which is extremely high here, this ratio. The nice thing about the test is they will get a summary page. These are wonderful for patients because patients see this above 127 when it's supposed to be maxed out at 10, and they freak out, right? Oh my gosh, this is so high. This tells them, okay, what's the physiological function of this ratio? It has to do with inflammation, right? It has to do with your ability to maintain cellular health and mitigate chronic inflammation. How does it get depleted? Well, it gets depleted because you're eating too many omega-6s or you don't have enough omega-3s because it's a ratio. How does this manifest, right? How do clinical manifestations deplete in the system? So you would be looking for cardiovascular problems, mental health issues. What are some food sources and what are some supplement options? So it really lays it out for you. Anything that is out of normal, whether it's yellow or red, will come with this type of detail for every single analyte that's out of source or out of range. So speaking of resources, you saw what was resourced or what was available as resources on the test, and that's very complete. But you also can have patient one-pagers, which you can have available in your office, which tell them, what is the test? Why are we looking at this? What is a deficiency? What are some things that I need? Just some like great overviews, but it's very patient-driven. So patient one-pagers available to you in your portal. You get key clinical messages, which is basically your Cliff Notes version of what I went through for these two tests. So you get one, I mean, it's available for all of our tests, but micronutrient, oxidative stress, it tells you all about all of that nitty gritty that you can, and markers and all of the information. So you can have like a little Cliff Notes version. We have interpretive guides. These interpretive guides are extremely complex or, comprehensive. They do have a lot of complex information in there, but it gives you what are the actionable items, right? These are like 43 plus pages to be able to say like, okay, this is the pathway, this is the backup, these are the nutrients, these are the lifestyle factors, and it just really lays it out there. So as you're learning, you can be able to really apply the information. There's lots of extra goodies in there. This medication interaction for micronutrients is probably one of my favorites because our patients are coming into us and they're on medications, and our medications are depleting our micronutrients in addition to our soils and everything else just being depleted of nutrients. So we want to make sure that as we see their medication list, we're talking to them about nutrients to be able to know that they're not kind of like falling into the trenches, and we're keeping them at optimal levels. There's some great webinars, micronutrients for athletes, for aging, all kinds of different things that you can look at. Once you start ordering the test and you are like, okay, now what, right? You can schedule a clinical consultation. These are free. You have access to our entire clinical team to be able to do these consults so that you can apply the information. You can tell them about the patient. They can look at the labs and they kind of reverse engineer, right? Because they see the labs first and then they talk to you later, and they kind of reverse engineer like what is going on with this patient and how does that lab relate to them. So a lot of people have asked, especially when we're in, I've met with several dentists, I've met with ophthalmologists, like, okay, well, I don't have a kind of a traditional medical office where we offer labs. Like, what do I do now? Well, you can have a traditional portal where you could have kits in office, or you can like place an order and have a kit drop shipped to the patient's house. We do have national phlebotomy databases and also can hook you up with mobile phlebotomy. But majority of our tests are available with an at-home collection option. And so just like the oxidative stress that I showed you, like this could be a drop shipped to your patients and then they can collect at home, and you don't even have to have phlebotomy in your office. You don't have to have a kit in your office. If you want to scan that QR code, you can open an account and your sales rep will contact you, and they will help you to design what is perfect for you, what works in your office. Do you want to have kits there? Do you want to have phlebotomy? Do you want to only offer the at-home collection? And if you want to try it out, there is a $400 testing voucher. So same QR code, you go to set up an account. As soon as you set up an account, you get connected with your local sales rep, and they will give you the code for $400 off, which means you can test, the two tests that I recommended today, you can get $400 off that section. If you really are like, I don't want to do this in my office, but I love the idea of testing, we do offer an e-commerce solution. So you can get a link that you can embed into your current website. It works very much like Fullscript, right? You can put in the tests that you want to offer. You don't have to offer our entire menu. You can choose like, I only want to offer micronutrient and oxidative stress. So when your patients go in, they enter their information. Those are the only two options. You tell them, I want you to order oxidative stress. They order it. It gets drop shipped to them. You can upcharge or add a service fee or add a consultation onto that. And then the commissions, all of that gets deposited right into your Stripe account. And there's also payment options for patients, which is really great because you sometimes in your office, and I know this, I tried to offer payment plans to patients, and it was like a nightmare for my front office and a nightmare for me. And most of the time, we didn't end up getting paid at the end. But there's a payment options and financing that's offered through the e-commerce site. So you get your money up front, the lab gets their money, and there's a third party that can handle that. And then your patient gets 3 to 4 months to pay over time, which can be really, really helpful when they're trying to, manage all different things. So just know that that is an option. It's same QR code. Just talk to your sales rep and they'll be able to help you set up with an account. So thank you so much for your time. I think we got right to the point where I'm about to lose my voice. So I'm going to take you off of screen share and we'll go over and check the Q&A. Okay. Let's see here. Okay, so we did have one question. Do we get any promotions for self-testing? We have one fee structure. Excuse me, hold on just a second. So there's only one fee structure. And then you can decide that would be considered what is the provider cost. And then you can upcharge on top of that or add consultation fee or however you want to kind of work that. And then you can do it through the e-commerce, but you can also just do it in your office. And then you have the promo code for self-testing for that $400 off. You don't have to use it on yourself, but treat yourself, use it on you so that you can really kind of see the benefit. Test kits available in Canada. Yes, we are available in Canada and across Europe. And so you can absolutely, I think the only yes, well, all throughout Canada, and there's some stipulations in, I believe, the Ontario, is that where Toronto is, area, as far as like how they have to be shipped in and out, but the company has already worked on all of that. So we have a dedicated sales rep, Brandon, who is in Canada. He knows all the ins and outs of all of that and can get you set up on all of that as well.
Right. I have a quick question.
Sure.
So I have obviously a lot of familiarity with your lab company. So what are some other subjects or other testing options outside of the few that you presented tonight? So you don't have to explain them all, but just kind of light touch on the other testing options that Vibrant offers.
Absolutely. So I think there are two of the biggest tests that we offer are going to be a gut zoomer, which is going to be our stool test. And then we also offer a total tox test, and the total tox is actually a bundle, and that is heavy metal testing, environmental toxin testing, and mycotoxin testing all together. So that really pairs nicely with the oxidative stress, especially if you have someone with a lot of damage markers and you're trying to figure out like how much is feeding into that oxidant bucket. Looking at their oxidate or looking at their total tox burden can be really helpful. And then in addition to that, we've got hormone testing, we have neurotransmitter testing, we have a organic acid panel. So we really run the gamut. We've got a lot of autoimmune tests. We have a whole tick-borne panel, which is the most comprehensive on the market. So we are striving to be kind of that one-stop shop for all of your functional medicine needs. And once you get in there, you can start to really explore all the different options.
I think we just got another question pop in.
Oh, good. Okay. Yeah. So how often do you test your patients for oxidative markers? So after initiating treatments, I like to test somewhere in that 4 to 6 month mark. And the nice thing about the test is when you order it initially, you order it as the genetics plus the damaged markers. And then when you reorder, of course, you don't need to order the genetics again. So you can just reorder just the damaged markers. So of course, that's a much, that's going to be a discounted price because you're only ordering part of the test, but it's still going to populate so that they can see that kind of previous and current. And I would say somewhere around that 4 to 6 month mark, as long as they are implementing changes and you feel like, okay, I have a pretty good handle on what were the things I needed to modify, and then at that point, now you have 2 data points. And then let's say that you have, your some are improving, but not everything's improving. Then maybe at that point you can dig a little bit deeper on the specifics of like running a toxin panel or looking for deeper for infections if you're not seeing the improvement that you think that you should.
A quick question that was out of the Q&A, Mexico. Do you offer the testing in Mexico?
Yes. Yep. Canada, Mexico, and Europe. There's actually only 18 countries that we don't work with. So majority of testing or majority of countries we will be able to ship to. And especially with our at-home menu, the things that are either dried blood spot, urine, saliva, stool, because they have a longer, stability to be able to get to the lab for processing.
And a couple of new questions for you.
Okay, sorry. Oh, I wasn't scrolling down. There we go. Let's see here. Is there a test that includes blood lycopene level? Unfortunately, not that I know of at this time. And then can you diagnose Lyme and co-infections? Yes, it is called our tick-borne test, tick-borne 1.0 or 2.0. Tick-borne 1.0 is going to be Lyme, tick-borne relapsing fever, and your most common co-infections, Babesia, Bartonella, Rickettsia. And then your tick-borne 2.0 is everything that's in that first part that I just said, plus additional co-infections, and some other opportunistic infections. So a lot of viruses and other things that tend to go along with somebody who might have a tick-borne infection. So we offer PCR testing as well as immunoglobulin testing, IgM and IgG for all of those tests. Okay, and how many snippets does your test check? I think you're probably talking about analytes. So it was 32 different SNPs. So the SNPs are going to be the single nucleotide polymorphisms. That's the genetic variation. We're testing 32. And then as far as damaged products, those are also our analytes, not genetic pieces, but the damaged products, we're testing 16.
Right.
Okay. Did I miss any? I don't think so.
Yeah, and we, if we get any questions, we could forward them to you.
Absolutely. Yeah.
Post webinar, it's a lot to digest, but really good information. So I'm sure things will pop up, especially when our viewers review some of the slides. And once we share all of that, I'm sure there'll be a lot more questions too. But
Wonderful.
yeah, this was great. And I hope the audience really found value with really approaching a new form of testing and understanding that there's more to it than a standard CBC.
Right.
And sometimes we can get much more information about the body by looking into functional medicine testing, and that's one of the purposes of tonight's webinar. So I don't think we have any new questions. So I just wanted to give a heartfelt thank you to Dr. Bruno, to Vibrant, and with this insightful webinar. I really think this is just the tip of the iceberg. We'll have more most likely, and your expertise and your valuable information shared today is really appreciated. So we hope everyone found this session to be informative, inspiring, actually, and in support of some of these modifiable risk factors. OWNS is launching out some things in the next month with our wellness guide coming out with a review of optometry. We have a downloadable guide for your patients, user-friendly for the Mediterranean diet, along with the ANA. So the goal is to really get your patients motivated to change their lifestyle and sometimes we need to show them what's wrong. So please don't hesitate to reach out to Owens, connect directly with Dr. Bruno's team. We look forward to continuing all these important conversations, and the recordings will be up in about a week or so. We will share as much as we can, and we appreciate everyone popping in tonight. It's a very testy night, but I do enjoy that we were able to proceed with this webinar. So thank you so much for your time, and thank you, Dr. Bruno.
Thank you. Thank you so much.
All right, everyone, I'm going to shut off everything in the next few moments. So thank you so much.
Thank you. Bye-bye.
Bye-bye.